So good morning guys and welcome to Excelsureacademy. Till now we were talking about the active transports wherein which we discussed about in the last lecture about the pore forming toxins, how exactly these can these toxins can cause the lethality to their host cells. Okay, so there is one more small topic like it's called ionophores. Now these ionophores is basically a kind of antibiotics only which are produced by the bacteria. So again these ionophores, they are responsible for, see you can see over here also two types are there, right? Again carrier ionophore and the channel forming. So you can see they also produce a protein carrier. They go and bind to these ions. Now when I say ionophore, it means we are just talking about over here ions only. Okay, so it can be Na plus, K plus or calcium 2 plus. So all these ions only, whereas in the other cases we talked about the molecules also, right? So there are two types are there. Either they go and bind to these ions and then, you can see over here how nicely they flip without exposing the charge to the phospholipid layer, correct, to the hydrophobic region. It doesn't allow to expose the ions to the hydrophobic region. Same way in the channel case, again it is totally completely closed and in between the hollow gap is there through which these ions are able to cross. Okay, so let's see, there are three channels are there, important ones. So I just collected those important ones which might come for the exam. Okay, so what are these ionophores? What are these ionophores? So they are hydrophobic molecules produced by the organisms that facilitate the ion transport across the lipid bilayer of the cell membrane. So two types are there, as just now we have discussed, the channel formers and the carrier compounds. So these ionophores make a hydrophilic pore in the lipid bilayer that allows the ion transport by avoiding their contact with the lipid bilayer. So just now we saw on the plasma membrane, they will be like a channel, so through which your ions can pass through. Now carrier compounds, these are the mobile ion carriers that facilitate the transport of ions through the lipid membranes by masking their charge. So on the plasma membrane, you have got something like this. To this your cation or anion binds, and then it flips and it delivers to the other side. Okay. Okay. So ionophores simply work as antibiotic and disrupt the ion gradient that is necessary for normal cell functioning. So this is what I was saying, that they act as the antibiotic. So remember the way we eat. So whatever antibiotics we take, can you name one antibiotic which we eat? Any name of the antibiotic?
Erythromycin,
Yes. Very good. Yes, Meher? So one I heard is erythromycin. Then, what else? Does paracetamol is an antibiotic?
Yes, ma'am.
And then yes. What about the others? Actually, paracetamol is not antibiotic. It is called antipyretic. Anyone who knows the meaning of antipyretic?
That substance to reduce the fever.
Yes, yes. So paracetamol is actually your antipyretic, which reduces the fever, or in other words, the temperature. Okay. Okay. So anyways, this is not right now important. So it is a potent act of defense among the microbes with their competitors. Okay. So as I mentioned, so now who are the competitors of microorganism? So we know that these microorganisms are freely surviving in the environment. So anyone can grow, but sometimes this area has been occupied by one type of organism. Okay, so what happens? So there are chances that the other microbes can come wherever the food source is available, all will try to come to that particular area with the help of chemotaxis. Now, when I say chemotaxis, anyone knows what is chemotaxis? Is it clear? Yeah? I hope it is not a new word for you guys.
Yes, ma'am.
So whenever food source is available anywhere, the organism use their chemotactic ability and they move towards the food source. So it's not that the only one type of bacteria or one type of microorganism will come. So many will come, correct? So when they all come, now there is a competition, like who can have the entire food source. That time they start releasing these kinds of antibiotics or the ionophores, depending on the condition, so that the other organisms will be killed. So with bacteria will have competition with bacteria, they can have with fungus also. Now when they infect us as humans, they infect us, so we are also food source for them, right? Because in our body also so many nutrients are available for them, so they attack us also by releasing on these ionophores. Now, as you know, superior being present on the earth, what we have done is, if I want to kill E. coli, I'll use ionophores produced by other species, for example, streptomyces, because they are the competitors. So what I'm doing, if I have infection with E. coli, I'm going to use ionophores produced by streptomyces. If I have with S. aureus, I'm going to use ionophores produced by the other organisms. So this is how the antibiotics have been, used, and their application has been studied, and it is there available in the market. Okay, so that's why you might have heard in your microbiology section broad spectrum antibiotic and all, right? So broad spectrum means one particular antibiotic is able to kill many other types of microorganisms. Okay. So this is again the same thing. So now let's just study about the few examples. So this is the table, and here I'm going to just let you know which are the important ones which you have to study. Valinomycin A23187, which is also called calcimycin. Okay, nigericin, gramicidin, and nystatin. So these you have to remember. When I say study means you have to remember for which they are responsible for the transport of the ions. Okay, so valinomycin is for K+. It is for K+. Did you know that valinomycin, like recently, has been declared to treat COVID-19 also? So it has got activity against SARS-CoV coronavirus. So I don't know whether it is there in the phase trials or not, but it was shown that the valinomycin is also having a good activity against the SARS coronavirus. I don't know how, because these are bacterial origin and our COVID-19 is viral, correct? So I'm not sure how both are connected and how they, claim to have this activity. Anyways, so just remember valinomycin is for K+, calcimycin for your divalent cations. Okay. So even the name says that it is related to calcium, please remember it is for divalent cations, where you have got calcium 2 plus Mg2 plus, or calcium 2 plus twice of H plus. Now gramicidin H plus K plus Na plus. Nystatin, monovalent cations and anions. Okay, so out of all these, just remember these 5, and a few of which we are going to discuss now. Please stop me in between if any doubts are there. Okay, so let's talk about the calcium ionophores. So you can see here the structure is also given. Don't worry, you don't have to study the structure. Nobody is going to ask you the structure. That's my guarantee to you. Okay, so it is also called calcimycin or A23187. Okay, so this is the name given by the company which has synthesized in the labs. Now, how does it produce in large quantity? It is through the fermentation by Streptomyces species. Okay, now if you see that whenever we talk about antibiotics, mainly it is from the Streptomyces species only that many of the antibiotics have been discovered. More than 1,000 antibiotics from Streptomyces species. Okay, so that much it is important. Now, they act as ionophores for divalent cations. Examples are calcium, Mn2+, Mg2+, strontium, barium 2+, etc., etc. Now, what is important about this calcimycin is how does it act as antibiotic is it uncouples the mitochondrial ATP synthesis activity. What does it mean? Yes, anyone who can tell me uncoupling the mitochondrial ATP activity? Can anyone tell me what does it mean? Anyways, in detail, we have to study all this, but yes. Yes, Ramya, do you know what does it mean uncoupling the mitochondrial ATP activity? Or otherwise, in simple words, does it inhibit the ATP synthesis activity or it promotes the ATP synthesis activity? What does it mean, uncoupling? Yes, Pallavi?
Inhibit.
Very good. It is the inhibiting, that is, it is stopping the ATP synthesis. So I'll just tell you in small, short, brief only. So during ATP synthesis from the mitochondria, as I mentioned about the electron transport chain. So from glycolysis, you get energy, then it goes for the TCA cycle, and then we talk about the electron transport chain, right? So in the electron transport chain, your mitochondria is playing the important role for the ATP synthesis, correct? So during ATP synthesis, what happens is it has been coupled with the what you call as the ATP synthase. Now what happens is uncoupling means it is inhibiting the activity of that. Coupling means the H plus needs to be transported towards the ATP synthase for generating proton electrochemical gradient, correct? Now this H plus proton electrochemical gradient has been disturbed, and because of which what will happen, there is no gradient means there won't be any ATP synthesis there. Okay, so this is inhibited by the calcium ionophores, for example calcimycin. Okay, so how does it act? So this is one example it has been given. So if I am using calcimycin for the treatment purpose, okay, like to treat, and it will result in the what to say, the production of interleukins. If suppose any mycobacterial attack is there, like for example in the case of tuberculosis is there, so how this calcimycin is going to help in the treatment against the tuberculosis. So this is one example is given. So by binding of calcimycin actually, so you can see that binding of calcimycin activates your NF-kappa B dependent pathway in macrophages. Okay, so what is NF-kappa B? What is NF-kappa B? It is your transcription factor. Okay, it is a transcription factor. Now, what is happening? It is activating your NF-kappa B. So calcimycin goes and binds to your P2RX7 receptor. Okay, you don't have to study all these things. Just understand how exactly calcimycin is inhibiting the tuberculosis bacteria from its attack. So calcimycin goes and binds to the receptor, activating your NF-kappa B. So NF-kappa B is present in the cytoplasm, bound with I-kappa B, where I stands for inhibitor. Okay, so what is happening? Activating means what? The I-kappa B will get phosphorylated. So once it is phosphorylated, it is free from the NF-kappa B. Now NF-kappa B is free. It goes into the nucleus and goes and binds to the gene which needs to get expressed. Okay, so let's say it is getting expressed for interleukins 12. Now this has been produced. So here there will be production of interleukins. Now interleukins will go and get released in the outside. Okay, extracellular will be there. Now what is happening? So it is what kind of this thing? Autocrine. Okay, autocrine signaling. It means the cell itself produces the signal, and the signal is going to bind its own receptor. So this becomes autocrine. Don't worry, slowly everything will study. So interleukins will go and bind to its receptor. Okay, now once it binds to the receptor, it is going to activate something called JAK-STAT pathway. Okay, now once this gets activated, it is going to increase the autophagy. Now what is autophagy? Self-eating, right? Apoptosis is self-suicide. Autophagy is self-eating. So when the mycobacterial tuberculosis goes and binds to the plasma membrane of the cell, it has been taken inside, okay, and this will be bound by your lysosome. It becomes your autophagosome and it gets degraded. Okay, and this is how your tuberculosis bacteria is being killed by the macrophages. Is it clear? I know it was little big, but I hope it was, I mean, I tried my best to explain. So should I explain once again? Yeah, so I'll just explain once again, okay? So your calcimycin, so we are studying how calcimycin treatment against the TB, tuberculosis. So calcimycin goes and binds to your receptor called this one, P2RX7. It goes and activates NF-kappa B. NF-kappa B is your transcription factor, okay? It is totally involved in all kinds of cellular signaling. Now, in the cytoplasm, NF-kappa B is present in the inactivated form with the help of I-kappa B, where I stands for inhibitor. Okay. Now, how it is activating? The calcimycin is activating by I-kappa B, making it phosphorylated. So I-kappa B gets the additional phosphate group. Now it is, see, whenever we talked about phosphate group attachment, there is always a conformational change. So till now, whatever the transport membrane we were studying, we always studied that whenever the ATP hydrolysis takes place, phosphate bind, there is a conformational change. Okay. Now, once the conformational change is there, the I-kappa B is not able to bind to the NF-kappa B. Okay. Now NF-kappa B is free. This NF-kappa B goes inside the nucleus, where it goes and activates the expression of IL, that is interleukins. Okay, so interleukins it will go and express. Now these are produced, they are secreted out of the cell because of the autocrine signaling, it goes and binds to its own receptor on the same cell. So we are talking about the macrophages, okay? So it goes and binds and activates your JAK-STAT pathway. So for the time being, just remember this name. We'll study in detail what is JAK-STAT pathway. Okay. It is actually called Janus activated kinase STAT pathway. Anyways, so it activates your JAK-STAT pathway, and that induces the autophagy. So what is autophagy? Self-eating. So when tuberculosis binds to the plasma membrane of the macrophages, it has been engulfed, okay, via endocytosis. Now this engulfed over here, this will be fused with the lysosome to become autophagosome, and finally call it as autolysosome, where we know that lysosome have got the hydrolytic enzymes, hydrolases, which will finally kill the bacteria present inside it. Okay. So this is how your calcimycin treatment is working. Okay. Next example is ionomycin. So now we talked about the calcimycin. The next one is about the ionomycin. So ionomycin is a membrane permeable calcium ionophore produced by the bacterium Streptomyces conglobatus. Okay. Now it facilitates the transfer of calcium ions into and out of the cells. It can be used to increase intracellular calcium levels triggering the cell death through the apoptosis and autophagy. So in the first example, in the case of calcimycin, we saw that it is inducing the autophagy, and here it is also through the apoptosis. So you can see how nicely it is actually a carrier mobile to which calcium binds and then it is transported. Okay, this is the structure of ionomycin. And now what is happening? It is actually changing the osmolarity of the cell, causing the cell to undergo dehydration. Okay, so once it goes dehydration, the cell will undergo apoptosis. So once again, so ionomycin is again a very good example of calcium ionophore produced by Streptomyces conglobectus. Okay, now they increase the intracellular calcium level and finally inducing the cell death through apoptosis and autophagy. Now coming to sodium ionophores, the very good example is monosin A. Okay, this is I don't know how many of you are from the agriculture background. So when we give feed supplements to the cattle like cows and buffalo, so you know that, I mean, the conditions are sometimes not that hygienic, right? The way we keep our food, it's not necessary that all the farmers they keep their the feed supplements for the cattle also hygienic conditions. So they are the grasses and all, they bring it from the farms, agriculture fields, they cut it and they just put it in front of the cattle feed. So there are chances that there are many, protozoa, parasites are there, which might, along with the grass, will go inside the cattle, right? So what will happen when these protozoa parasites are there? So they also start getting the stomach infection, they start getting the bloating, and then it is difficult for the cattle to digest the food. Okay, so in these cases, these antibiotic that is called monosin is given. Okay, so it is the antibiotic which is isolated from Streptomyces cinnamonensis. It is widely used for ruminant animal feeds. Yes. So it is extensively in the beef and dairy industry to prevent coccidiosis. Okay, please remember this word, coccidiosis. So which is the increased production of propionic acid and causes bloating in the cattle. Now, they transports monovalent cations such as lithium, Na+, K+. So there, when we talked about calcimycin and ionomycin, it was all about the divalent cations. Whereas here it is also, in this case it is monovalent cations. Okay. Now, monosin A is able to transport these cations across the lipid membranes of cells in an electroneutral exchange, playing an important role as a Na+, H+ antipoter. What do you mean by electroneutral? Also said as non-depolarizing exchange. What does it mean? What is exactly happening? Anyone? Yes? Akshita, any idea what is happening? What do you mean by electroneutral exchange? So electroneutral exchange means that when you have got the channels for the ions to be transported, if Na+ is going outside, inside H+ will come. So the charges has been cancelled. So if one positively charged ion is coming in, one positively charged ion is going out. Okay, so it means it is a antipoter. So that is the meaning of electroneutral. Now, when we were talking about the calcium, we just mentioned that it is just transporting the ions in one direction. So there it is not electroneutral, because in one side of the plasma membrane, the charges are going to increase. Whereas in this case, it is not. So if two charges comes intracellular, two charges are going out also extracellularly. Okay, clear?
Ma'am, what is bloating, ma'am?
Bloating, our stomach, you feel gas is there, na? Sometimes we say that, oh, I got gas, and your stomach, it increases and you are not able to digest. Your stomach actually
Okay.
becomes full of gas, acidity and all. That is bloating.
Okay.
Huh? Okay. Simple diagram. So where you can see that these ionophores are there. So calcium goes from your gut lumen to the parasite. Okay. So these parasites are present in the ruminant gut. It means what? In the gut of the cattle. And then they actually cause this exchange and kill the bacteria. Oh, sorry, kill the protozoas or the parasite. Now talking about the potassium ionophores. So as I said, this is very important. Valinomycin. Okay. So it is a natural neutral ionophore. Again, when I say neutral, it means it has got no charges. Okay. Obtained from Streptomyces sumimensis and Streptomyces fulvicimus. Okay, I'm so sorry, you have to remember all these words, the name of the bacteria. Okay, it is important. Yes, it is a dodecadepsis peptide highly selective for K+. Oh my god, what does this word means? Any idea? It says that there are 12, okay, and in peptide bond we have. Okay, so whenever we talk about proteins, we talk about one single peptide bond having one carbonyl and amide group, right? When we talk about DEPC, it means that it has got two peptide bonds. Okay, so two peptide bonds are there. Now, it has recently reported to be the most potent against the SARS coronavirus, as I just mentioned before. Now, look at the structure over here. So, in this case, you have to remember the basic sequence of the structure. Okay. So, what you have to remember here is that, see, the structure, what you have to remember is that it has got L-valine, dihydroxyisovaleric acid, D-valine, and lactic acid. Okay. So, 4, 3, 12. So it is dodecacapsy and it is Jepsy because of the presence of the double peptide bond. So what you have to remember is not the structure but the sequence. L-valine, hydroxyvaleric acid, and D-valine and lactic acid. Okay. So this ionophore has got amino acids of both conformers, L and D. Remember, I have mentioned that naturally occurring all sugars are in D conformers, whereas all amino acids are in L conformers. But only in the case of bacteria, you can also see the presence of D-valine. So in the coming lectures, we'll study about the peptidoglycan also, in which your D amino acids are present. Okay. So these are few exceptions where we have got the naturally occurring D amino acids instead of L. Yes, is it clear this much all about the ionophores? So we discuss about the calcimycin, ionomycin, then monensin A and valinomycin. Okay. Now question time. So I got very good questions so that your brain will become active now. Yes. So the first question is given. The parietal cells of the stomach contain membrane. So parietal cells means which produces HCl in the stomach, okay, which induces your hunger. Yes, this you people have to solve. Okay. So they contain pumps that transport hydrogen ions from the interior of the cell, that is pH 7, to the surrounding medium to make gastric juice pH 1. So we know that whenever there is a hunger signal is given, the acidity of the stomach is increased. That is from pH 7 to 1 it has been made. Okay. Now calculate the energy required to transport 1 mole of hydrogen ions. So first of all, which formula are you going to use? If you guys are listening to my videos and lectures and noting down, then you should remember the formula. So I have given you the intracellular and extracellular, that is in and out, the concentration of the H+. So you need to calculate the energy. And that too, it is given that it is a free energy. So free energy means what exactly you have to calculate? Someone has to say something, otherwise, anyways, the timer is also started. The meeting will get over in few minutes. Yes. Okay. So how you have to do is, we know that the free energy we need to calculate. That is called delta G. Delta G is equal to minus RT ln C out by C in. This can be written as minus 2.303 RT log, right, C out by C in. Also, this can be written as 2.303 RT log C in by C out. I have taken the minus sign and converted these. Okay. So I want the answer in positive value. So I can do that also. You can just, make the numerator as denominator, yeah, and take the minus sign out. Now let's calculate. So what are the values given? So we know that it is equal to 2.303. So what is the value of gas constant? So the value of gas constant is 1.98 calories per degree Celsius. Okay. Since they have not mentioned at what temperature, so we are going to take the normal physiological temperature, that is 37 degrees Celsius, but it has to be in Kelvin. So what you have to do, just add 273 and you are going to get how much? No, it is not 37 degree. We take as 25 degree. 25 degree it is taken. So what you get is actually 298 when it is not mentioned. Okay. Then we have got the log 7 and 1. So this becomes, so this is what I have just converted, right? So this becomes pH1 means what is the H plus concentration? 10 raised to minus 1. Okay. Now if I say H plus concentration is pH is 7, it means it is 10 raised to minus 7. Okay. So this is how you write the H plus concentration. Now I have the values. I can put 10 raised to minus 1, 10 raised to minus 7. Now when I solve, I get 2.03 into 1.98 into 298. So how much I'm going to get? Log 6. Correct? Now when I solve, so when you solve everything, so log 6 is how much? Log 10 raised to 6 is 6. Okay. This is basics logarithms, you should know. So what value you are going to get? You are going to get as 8.2 kilocalories per mole. Yes? Was it difficult? No, right? It was easy. The only thing is you have to be very quick when you do the multiplications. And every time you don't have to take the entire values. You can just take the starting 2 digits and just write the answer. Because in IIT-JAM, they will ask you the near correct 2 decimals. Okay, so when I solve this thing, for example, I got as 8150 as calorie per mole. But if they're asking in kilocalories, so what I'll do, I'll just round this number and I'll write it as 8.2 kilocalories. You can write this also, that also, since it is a numerical based written. So they will see your steps and everything. So did you see that it is actually taking only 2 line calculation only, and that much only space will be given for your IIT-JAM paper. So if you have seen the question paper, one question is given and only this much space is given. So you should know what exactly to write. You cannot just write, let us assume that this is and that. Okay. So is it clear? Let's move on to the next. Okay. So the next question is not numerical, but you have to think. So we know that glucose is frequently administered intravenously to the patients as a glucose source, right? As a food source. So whenever you are unconscious, so glucose has been used. So IV, the drip is there, it is having the glucose, but in the packet it will be written as dextrose. So don't get confused. So given that the transformation of glucose to glucose 6-phosphate consumes ATP, why not administer glucose 6-phosphate directly instead? It's a good question, right? Why you are making your body to convert glucose to glucose 6-phosphate? It is better to give glucose 6-phosphate directly. So what will be the answer for this? Yes, fast. Yes, Ranjan, what do you think? What will be the answer?
Glycolysis, I think.
That's okay. So even if I give glucose 6-phosphate, that is the second step in the glycolysis. So it can continue the glycolysis. No problem with glycolysis. It is related to the transport membrane till now what we were discussing. That is the only hint I will give you. For those who don't know glycolysis, so we have got glucose, then glucose 6-phosphate, then fructose 6-phosphate, right? So instead of giving, starting from this step, if I give glucose 6-phosphate directly, it will also continue with the glycolysis. So there is no problem with the glycolysis. It is something else. What it is? Something related to membrane transport. Think. What is happening from glucose to glucose 6-phosphate?
Phosphorylation.
Correct. Then what else? Because of the phosphate group added to the glucose, what is happening to the glucose? I'll make you think and give the answer. So from glucose,
Phosphate is adding.
yes, phosphate has been added. Agree. 6 is the carbon group to which your phosphate group has been added. But what happened now?
ATP is added.
No. Phosphate group is added, ATP is used.
ADP is released.
Oh, Baba. I know all that. I'm asking what had happened to glucose with respect to its structure, with respect to its function. What happened to glucose now?
Phosphate group is added to glucose, ma'am.
Arey Baba, I know that.
Permeability.
Ranjan, what?
It may be permeability.
Yes, now he got the point. So what had happened? Glucose has become glucose 6-phosphate. It means what? From this size, it has become this size. There is an increase in the size of the glucose molecule. Do you agree, everyone? So there is an increase in the size plus phosphate group is there. It means what? It has got the negative charge. Now what I taught about the permeability of the membrane. First of all, glucose is a polar molecule. Now you have increased the size also of the glucose. So the size has been increased. There is a negative charge. Okay. So now it becomes impermeable to the membrane. It is not able to cross the membrane. And for glucose to transfer, we have got the GLUT transporters. Now, if I give glucose 6-phosphate directly, it is not going to detect the GLUT transporters. It will not bind. Moreover, it has got the negative charge. Again, it will not cross the membrane. So because of this, what is happening? It will remain outside the membrane only and present in the blood. It will never be taken up by the cells. Plus, so this is why we don't give glucose 6-phosphate. We always give glucose so that your body, your cells are able to convert into glucose 6-phosphate. Okay? Now it's clear. One more thing. In the once the glucose is inside, immediately it gets converted to glucose 6-phosphate. Okay, and this is the reason why even if the concentration of glucose 6-phosphate increases, it will not go outside, because outside glucose concentration is high, inside glucose 6-phosphate concentration is high. So here the solute osmolarity is not being hampered, and because of this, once it gets converted into glucose 6-phosphate, it cannot go out of the membrane. If suppose glucose was inside also and outside also, then it will try to become into the equilibrium condition. But no. So nature has made our body cells in such a way that it immediately gets converted into glucose 6-phosphate, and it cannot go outside. Okay, otherwise it will be like, you know, equilibrium between the glucose in the blood system and your body cells. So in order to avoid that, the moment glucose is there, it is taken inside of the cell, it gets converted to glucose 6-phosphate. Okay, now why we do not administer glucose 6-phosphate? Because first of all, the size had increased, plus it has got the negative charge. It becomes impermeable to the membrane to cross, and hence we don't give glucose 6-phosphate, we give glucose. Okay, now it's clear.
Yes, ma'am.
Okay. So thank you for listening, and I hope you really understood everything. I hope we should do more of such questions, right? Chalo, thank you.